Great. Okay, so Dr. Meghan Azad has selected our second 2024 Canada Gairdner Early Career Investigator, and that is Dr. Tsai. Dr. Tsai is a Canada Research Chair in Immunometabolism and Diabetes and an Assistant Professor in the Department of Medical Microbiology and Immunology at the University of Alberta. She completed her PhD in Dr. Per Santamaria's lab, where her thesis focused on tolerance induction in type 1 diabetes. She then completed postdoctoral training with Dr. Dan Weiner here at the University Health Network in Toronto, and during that time studied mechanisms of immune dysregulation in models of obesity-related insulin resistance. Using a combination of animal models and immunological approaches, her lab studies the role of immune metabolic crosstalks in diabetes, infection, and cancer, and explores potential new strategies to improve immunity. Dr. Tsai will be presenting maternal humoral factors modulate offspring gut immune homeostasis to mitigate diabetes development. Please join me in welcoming her to the stage.
All right. Thank you for that kind introduction. It is a huge honor and also a humbling experience to share the stage with the laureates that pioneered the technology that we rely so heavily upon in my lab. And it's also inspiring to speak after Meghan, who provided such important translational aspects of maternal factors and breastfeeding research, which is so relevant in so many chronic diseases, including the ones that my lab is passionate to study. So as Meghan has beautifully explained, breastfeeding and early life exposure to maternal factors play a huge, profound, and long-lasting role on an individual's resilience to diseases through shaping their microbiome, through shaping their immune development, and this long-lasting effects persist long after the withdrawal of these initial early life maternal influences. So this is really an important field to study, and one good example of a disease that is altered by maternal influences is type 1 diabetes. We know that type 1 diabetes, or kids that are born from mothers with type 1 diabetes, they develop resistance to disease compared to kids that are born from mothers without type 1 diabetes, suggesting that the affected mothers are conferring protective factors to these babies. But to this day, we don't fully understand what are the mechanistic underpinnings of this protective mechanism that are provided by the moms. Type 1 diabetes, for those immunologists in the audience, is an autoimmune disease. It is mediated by autoreactive T cells, CD4 and CD8, that recognize peptides expressed or presented on MHC by antigen-presenting cells, and these are peptides that are expressed by beta cells that produce insulin in our bodies. So what happens then is that these T cells can home to the islets and mediate beta cell destruction, and that results in patients that become lifelong dependent on insulin injection to maintain their glucose homeostasis. This disease is shaped by genetic factors, but we also know that there is a heavy influence from the environments. And if we look at these environmental triggers or modifiers of disease development, you can see that many of them fall within this early life period, so during the prenatal or postnatal stages. Many of these have to do with infections, diets, mode of birth, nursing, and we know that all of these factors have one thing in common, and Meghan had elaborated on this, which is the microbiome. So all these factors can contribute to shaping the microbiome in individuals. So in type 1 diabetes, we know that dysbiosis, or an imbalance in the microbiome in the gut, can impair intestinal barrier function, and through that allow bacterial products as well as metabolites to increase their translocation, gain access to the immune system, and aggravate autoimmune responses. But we also have built-in protective mechanisms, and one of them being this green dimeric antibody here. This is IgA, and we know that IgA can bind these bacteria in multiple ways to restrain them and protect against dysbiosis. So what IgA does and how it's linked to type 1 diabetes is not entirely clear. We know that in patients with type 1 diabetes, there's an increased incidence of what's called IgA deficiency. So this is characterized by low serum IgA, which is different from the mucosal dimeric IgA. But we also know that the fecal IgA, they bind bacteria in a way that's very different when we compare patients to healthy controls. We also know that IgA is the predominant immunoglobulins secreted into the gut as well as the breast milk. So this led us to ask the question, what is the role of maternal IgA in type 1 diabetes? To answer this question, we had to make an IgA knockout mouse. This was 6 years ago when I was a postdoc, and I convinced my supervisor to let me breed mice in the facility here on the 6th floor. And we spent 2 years making this mouse, and then in my lab, when I started at the University of Alberta, we started following these mice for diabetes. But to answer this specific question, we had to do a very specific kind of animal breeding. So we had two breeding strategies. We generated heterozygote pups, so they are heterozygotes for IgA. But in one cohort, we had these pups that are born and nursed, or birthed and nursed by IgA sufficient dams, so they would have received maternal IgA for the first 21 days of their lives. And on the other hand, we have pups that are genetically identical to these pups, but they did not receive maternal IgA during this nursing period. But note that both pups are capable of making their own IgA. So we spent 32 weeks to follow diabetes incidence because that's how this model works. And as you can see, these mice develop spontaneous type 1 diabetes because of their genetic predisposition. But after following all these mice, we proved ourselves wrong. We thought that maternal IgA deficiency would exacerbate disease. But to the contrary, we found that maternal IgA deficiency protected the mice against type 1 diabetes. We looked at the immune responses in these mice and the phenotype using tetramer staining and tracking for autoreactive T cells showed us that these mice are indeed, they developed diminished autoreactive T cell responses to islet antigens and were protected. So at this point, we were disappointed because we were a startup lab and we didn't know where to go. But then we kind of put our feet down, and then we decided we need to figure out what is protective that these dams are giving to the pups, because that could be beneficial in terms of thinking about the early life influences and how that modulate diabetes development. So we pressed on with our project, and we keep doing experiments to try and understand this relationship better. So one of the experiments that we did was we did a cross-fostering experiment where we swapped the little pups at day 1 of age onto either a wild-type IgA dam or a knockout or IgA knockout dam. And what we found was that just by being nursed by IgA deficient dams during this early life 21-day period, we could recapitulate the protection that we saw in the maternal IgA deficient pups. So we concluded then that maternal protective factors are transferred during nursing. So we know that during nursing, the dams can provide the pups with either milk or microbiota. We're following on the microbiota, but I want to share our data on the breast milk study because it was really a challenging effort to mechanically extract milk from mice. You can ask me about this later. But my student, she prevailed. She got enough milk so that we could do proteomic analysis of the 2 different milk samples, and we were able to identify more than 3,000 peptides, and a lot of them were very different or up or down regulated in our IgA knockout milk. And you can see highlighted in purple that many of these proteins that were altered were antibodies, so echoing to Meghan's talk on how important that breast milk antibodies are in shaping the disease development in the pups. So we did then next look at the antigen specificity of these breast milk. We looked at what they were binding in terms of the fecal bacteria communities. And to make a long story short, what we found was that the milk from the IgA knockout dam, the antibodies within that milk are much better at binding specific strains of bacteria, and those strains of bacteria are suppressed in the corresponding pups, suggesting that there is an interaction between maternal milk and the gut microbiome of the pups that could be involved in diabetes protection. So we are still following on that to see if we actively induce immunity to these strains of bacteria, can we then protect the pups from developing diabetes? We also looked at how this altered microbiome and maternal breast milk composition altered or impacted the gut environment in the pups. We did RNA sequencing, and we saw an upregulation of pathways that are involved in antimicrobial resistance, defenses, pro-inflammatory pathways, and we validated these with real-time PCR. And so there's clearly more inflammation in the gut of our protected pups, and this was at 3 weeks of age, and it also went against a little bit what we predicted would happen. We looked at the immune composition in the gut tissues, and we saw that the gut T cells were more activated. We saw an expansion of IgA plasma cells, and importantly, we also saw an expansion of induced regulatory T cells in the gut. And when we compared the mucosal barrier of our protected versus non-protected pups, we saw an increased thickness, a more robust mucin layer in our protected pups compared to the ones that were not protected. And so this is what we think is happening, although a lot more work needs to be done, but we think that the maternal IgA deficiency alters the microbiota, which interacts with maternal humoral factors to induce a strong inflammatory event during weaning. This is what's called a weaning reaction, and that leads to beneficial imprinting of the gut, bolstering the barrier function, expansion of regulatory cell types that then result in the suppression of autoimmune diabetes in the islets. So I want to end by echoing back again to Meghan's talk to say that timing is really everything, and we still have a lot to do to understand the maternal factors and how they influence the development of type 1 diabetes. But we really truly believe that this early life period is crucial and is a window of opportunity to develop prophylactic strategies to curb this disease. So I would like to thank my lab. Erin was the one that developed the milking and most of the work in this talk, and also my other trainees, my collaborators, and also my mentors and funding agencies, because without them, this work would not be possible. And thank you.